Okay, good afternoon, everybody. Thank you for joining us after lunch. We will try and be energizing as a panel, so to stave off the lunch lethargy. So this afternoon, I'm joined by three fantastic panelists, where we're going to be discussing, I think, a critical item in the conference, which is how do we regulate a damage repair approach to aging? So we've learned throughout the last few days about the exciting science that's going on. We've learned about the patient advocates that drive the need, and we've also learned about the investment paradigms as well. Today, what we're really going to be focusing on, again, as always in my panels apparently, is the lab-to-bedside approach. So we're going to kick off firstly with Evan Schneider from Stanford Burnham, who's going to talk to us about some of the underpinning science which may be used to treat some age-related diseases, and thus the regulatory implications of them. Then it's been followed by Andrew Martello from Spoonful of Sugar, which is a company which looks at novel patient adherence strategies. So when we've got a product, that's great, but how can we incentivize and monitor patients to actually take these therapies to maximize efficacy? And finally, we're going to hear from Bob Clay, Chief Regulatory Officer from Kynaps, and he's going to tie together the ends which Evan and Andrew create, but also offer some perspectives from his experience in many years in the regulatory industry of how we can best deal with the challenges which are likely to be posed. So thank you very much for joining us. And to be clear here, for those of you who are planning lunch breaks or calls, we're going to go through until 1:50. But synchronize your watches if you must. So without further ado, Evan, stage is yours.
Thanks. Well, this is kind of interesting. I just ended the last panel, but as promised, I'm going to reiterate a lot of the points. In fact, I'm going to show almost the same slides that I showed at the previous panel, but this time what I'm going to do, that one was talking about a lot of the biology. What I'll do is, besides fleshing out the story, because I have a little bit more time, is in each indication, I'll talk about what the regulatory implications are of some of the biology that I was talking about, just to be apropos to what to the theme of this session. So this sort of came out during the panel discussion, though not during my talk, but from the earliest days of the stem cell field, and I can attest to having been there, these are the kind of things that we were always asked. In fact, this is a cartoon that was, I think, published in The Guardian when we first published the very first human neural stem cells. And I don't want to tell you how long ago that was, but that was the joke, that we're going to do this. And I put it off, and from that very beginning, and that's almost 20 years ago, people wanted to know, so when are you going to do this? And, you know, over the years, it's gotten closer and closer, so that now some human neural stem cells are in trials for some diseases. But my answer always used to be, when are we ready? And I would apply this 5-point test to anything when I was asked this question. One, I would say for the given entity, is the standard of care suboptimal, or is there no therapy or cure exists? Then the single most important thing that I would always ask was, does it make sense? Does the biologic data make sense based on our knowledge of the cell's biology and based on our knowledge of the disease's pathophysiology? And that would be the deal breaker. If it didn't make sense, then it was not going to advance either scientifically, and it would definitely not make it through a regulatory gauntlet. And then these other things were, of course, if it's done, are better proven therapies not jeopardized. So, example, we were doing a lot of stem cell work for brain tumors. We had to make sure that our experimental therapy still wouldn't deprive a brain tumor patient from getting radiation, which still is the only thing that shrinks a tumor. Is it safe? Obviously. And then, does it do no harm? And that includes financial harm, psychological harm, ethical harm, if you're thinking about that. So I think in the previous session you heard Julie talk about all the various aspects that you are concerned about with regulation. For me, and I think I had also mentioned that I chaired the FDA's advisory committee on cell, tissue, and gene therapies. And despite all of the things that my committee was most concerned about when we were presented with something, whether it was the results of a clinical trial or having to come up with standards, was what is the mechanism of action of what you're proposing in a patient and in a pathological environment. And it was recognized this is a moving target. It's always changing. The complexity of the disease changes, and I'll get to that, which means our growing sophistication of what the cell's action is keeps changing. So yes, there's a moving target in terms of what your release criteria are, what your assays are, what your targets and goals are. But it's very important. But from that's where everything else moves. Everything moves from MOA, mechanism of action, to proper POC, proof of concept. Is it better than SOC, which is standard of care? Is that your potency assay? And based on that, you constantly change your criteria. So you're not meant to read this, but after you've done that, then all these other aspects of manufacturing, then you can focus on that. Because you can't imagine, this is a lot of work and a lot of money. And if you're going down the garden path, then you've just wasted a lot of time and a lot of resources and raised a lot of hopes unnecessarily. So again, to reiterate, a mechanism of action, proof of concept, and then clinical competitiveness. Stem cell therapies, they're cool, they're very elaborate, but they're very expensive, often invasive. At the end of the day, is it better than what exists right now? Now, for many diseases, the answer is clearly yes. There is no cure and these are lethal. But for others, for example, maybe in the musculoskeletal field, if what you want to do, even with the coolest chondroblast, if it still is no better than bed rest or some anti-inflammatory agents, then it doesn't have clinical competitiveness. So it can be cool, but it's not going to be a winner. So now, let me go through this list that I did earlier in the, that I did in the morning session about looking at everything that we've talked about over the past week that we think is in the, is in the armamentarium of rejuvenation biomedical industry. And it's moving from probably the kind of easier to really, really hard, or at least easier in concept. And let me go to some of the examples that I gave before. So what we want to do, and this came out, and I'm going to spend some time on this. This came out in one of the Q&As, if you were there, that ultimately the best intervention for the diseases of aging is prevention, anticipation, predicting, and preventing it from actually happening. Rather than than digging yourself out of a hole, see if you can prevent digging the hole to begin with. And the best way to do that is going to be personalized medicine, meaning by personalized medicine, having a bank, a database of every individual's molecular and functional, meaning cellular organism makeup, archived and readily interrogatable throughout life for early intervention. Now, first of all, how are we going to get this going? But in a way that I wasn't able to flesh this out before, why would you want that? I think it becomes obvious, but let me articulate it very clearly that up until this point, most therapies are trial and error. They are not drug choices. Whether somebody gets surgery or not are rarely based on a patient's own pharmacogenetics, prediction of whether he or she is going to respond or not, prognostic indicators, which means that's expensive, time-consuming, and risky because while you're putzing along, this pathologic process continues unabated. So it also is risky, and then compounded by off-target and undesirable side effects. Personalized medicine would bring evidence-based rationality to the intervention. So it optimizes the chance of efficacy, minimizes the chance of adverse effects, and ultimately diminishes cost. And one way is it's unresolved. How do we do this? Now, George Church has one particular approach, and he's really been a leader in trying to make personalized medicine concrete and real. And he's doing it from the aging end. I'm a neonatologist and a pediatrician, and I'd like to think that we're starting to approach it from the other end, and maybe we'll meet in the middle, which is the fact that every baby has an umbilical cord. And it gets discarded. When the baby's born, it goes into the trash, but you capture all kids, and it's a sample of that baby, his genes, his cells, and this indicates that we've learned how to, in a very high throughput manner and quickly, turn them into HUVECs and then IPCs in a very efficient high throughput manner. So now, what the speculation would be, we can now correlate what's going on with this baby's cells and his genome and correlate it with what goes on in his life as he moves forward. The conceit, of course, is that if we profile this non-invasively, that we will improve care, particularly if we recurrently and prospectively profile and interrogate. In the NICU and in ICU, anybody who's ever been there, every aspect of these babies is monitored. If they hiccup, it's recorded. You know every drug, you know every heartbeat. Going forward, these kids are followed, first by the pediatrician, and then when you're an adolescent, and then when you're adult. And the conceit would be that going forward, we have this database to constantly interrogate. Is this a kid who winds up developing penicillin allergy? Does he wind up developing a brain tumor? Does he wind up developing diabetes? Does he get autism? Does he go on to develop Alzheimer's disease or Parkinson's disease? And the notion would be, either because we've done it, or I'd like to partner with some umbilical cord banks, because the capture is huge, that we do omics. We constantly correlate this with clinical outcomes and family histories, back and forth, a heavy bioinformatics, and then we perturb with known perturbogens and stressors and see how the patient responds. And this becomes the patient's personal molecular cellular history, personalized medicine, if you will, pathway analysis. But then ultimately, for the sake of science, we develop biomarkers, validated biomarkers, and that's the way that rejuvenation medicine really works, because now you anticipate problems, which was one of the points of the Q&A. And of course, you have a personalized bank of everybody's cells. So as I said before, I'm looking for, if people are talking about blue sky, and Peter in this morning session talked about what's a big idea. Well, this is one. So I'm looking for clients, I'm looking for partners, I'm looking for investors, I'm looking for donors to actually make this happen, because this is low cost, inexpensive, personalized medicine, bringing it to the masses. So predicting, which means, of course, you have to know what cell type you want to look at, and that can vary. If you're taking them from iPS cells, they may not be mature, and if it's a disease of aging, that could be a problem. And it is known that some cardiomyocytes, for example, do not manifest a disease until they've been metabolically aged. Even when they come from a genetic disease, then you see it. So that's a caveat. The other caveat is that pure cell populations may not recapitulate a disease. And this morning I talked about our experience with ALS, where initially we thought that getting motor neurons was our goal. This was a study that took longer than I care to admit. It was a decade and a multi-center trial. When we started out at the beginning of the decade, this is what we wanted. By the end of the decade, what got us long lifespan extension and functional recovery in these animals was not motor neuron replacement, but recognizing that the astrocytes were toxic, so impeding them and replacing them with good, healthy, trophic factor supporting protective astrocytes, and then some gray matter oligodendrocytes that were also protective. And then knowing what do you want to profile? If you want to do personalized medicine for predictive value, what do you want to profile? And the profiling is not just the genome, but also what actually drives the cell, which is the proteome. And then assaying for clinically meaningful predictors. Just because you see something in a dish doesn't mean that that has anything at all to do with the symptomatology of the actual disease. So looking at Rett syndrome iPS cells in the neurons, one can see changes in spine density. Nobody knows, that's interesting, but nobody knows whether that has anything to do with that kid's autism. And if you screen against spine density, maybe that'll be the wrong thing to do. Now, at least with monogenic diseases, we have some kind of handle, something, and often a cell type. The challenge, as I talked about before, are these complex diseases, multifactorial diseases. And then I talked about how for those, what you want, even if you don't know the mechanism that something works, you want to kind of do what we call a molecular can opener. And neuropsychiatric disorders are the poster child of these kind of diseases that have no gene, they're polygenic, multifactorial, very complex. But a molecular can opener is an example would be in bipolar disease. For whatever reason, nobody knows why, half the patients respond to lithium, but we don't even know the target. But if we get inside the cell, what we're able to do was find lithium's protein target in the cell. It turned out to be a molecule that's a fundamental developmental molecule that's druggable. It's druggable because it's based on its phosphorylation state. It's also very critical in dendrite development, but it's also quite responsive to external cues. And I guess in the interest of time, I'm not going to go through all the details, but to say that it's a molecule called CRIMP2. And what just seems to be the case that when it gets phosphorylated, neurites fail to grow. There's poor connectivity, poor neuronal function, and it just seems to be a little bit too high. The set point is too high in these bipolar patients. So what lithium is brings down the set point. Well, that's kind of interesting. And what we've been able to find is that, again, in the interest of time, that it works in models, animal models, to the best of our ability of bipolar disease. In other words, animals that are treated with methamphetamine, which is a model of bipolar disease, and wild-type animals become absolutely nuts. They don't stay. They keep circling the perimeter of a cage, whereas the animal whose crimp cannot be phosphorylated, so now we have a target, is like he's on chronic, it's as if he's on chronic lithium. But once we had that, now it starts getting a little bit interesting. This is the mechanism. When it gets phosphorylated, it releases all these cytoskeletal elements. Now that starts linking this disease and development to Alzheimer's disease, because that's exactly what seems to happen in these Alzheimer's plaques. And now you have Alzheimer's plaques. There's other tauopathies that are not diseases of aging, like chronic traumatic encephalopathy, these contusions that NFL players get and that the troops are getting after concussion. But it's the same kind of pathology, and we think it's linked to that. So now we have a drug. Where's the regulation here? It seems like it's pretty that personalized medicine should be pretty straightforward. And in fact, it's not even invasive. Well, where this comes in is how to protect confidentiality of the patients, what happens when intellectual property is developed out of here, a drug target, a diagnostic, a prognostic, maybe even the drug itself. And one way to deal with that is maybe let the patients be stakeholders and let them hold some of the IP. I want to go quick. I probably only have about another 5 minutes. I'm going to go quickly through some of these other aspects from a regulatory point of view. We talked about preventing and intervening. That, to an extent, it means using personalized medicine and then doing high-throughput drug screening and looking for targets. For what I was just telling you before, in this whole pathway for bipolar disease and maybe for other tauopathies, once we knew what this was, then we could figure out the pathways. And once we knew the pathways, we could figure out all these other kinds of drug targets, ways other than lithium, because lithium, anybody who knows anybody's been on lithium, it's no day at the beach. You want to get rid of lithium, but you want to lower phosphocrimp, for example, and there are other targets here. Interestingly, some of them are FDA-approved drugs that can now be repurposed. What we found were 2 cancer drugs that aren't doing their job in cancer, but they are lowering phosphocrimp. So in terms of regulation, you've dodged a lot of bullets. If you can repurpose an FDA-approved drug, meaning it's already gone through safety, that's a winner. And that's something that would be great to be able to do, and that comes out of personalized medicine and prevention, and this is another way of preventing things. Protecting what we had, and I mentioned that this discovery in the stem cell field actually came from looking at a disease of aging. It happened to be Parkinson's disease. But the notion is that cells have a division of labor, neural stem cells, because they're there actually to participate in organogenesis and homeostasis. There's a division of labor between the cells, and one of the jobs, this guy might become the missing cell, but this one is a chaperone cell. And it chaperones the host as well as this cell through diffusible factors, through cell-cell contact, and now we're starting to learn even through exosomes and microvesicles. It thinks it's just maintaining homeostasis. We co-opt it for protection. And that's what worked in a monkey model of Parkinson's disease, as I spoke about this morning, that neural stem cells into a monkey model, which is an authentic model of human Parkinson's disease, had an effect in lowering Parkinsonian features, not through one mechanism, but multiple mechanisms. Dopamine cell replacement was actually the minor contribution. The major contribution was preservation of the nigrostriatal pathway, as illustrated over here, and also the cells cheek by jowl, the green cells next to the red cells. The green cells are the neural stem cells, the red cells are the dopamine neurons, protecting these guys, and then also helping to eliminate alpha-synuclein aggregates. It was a big part in ALS. I won't have time to go through all the data, but here are the Kaplan-Meier curves. This is extensive survival, the largest survival ever reported for the ALS animal, but we had nothing to do with this, really. You know, we thought we were replacing motor neurons, but we weren't. They didn't go out the ventral roots, they didn't make connections in the neuromuscular junction, but what they were doing was cranking out tons of GDNF, and if we put them right in the key areas that sustain life, the respiratory center, grooming, and things like that, then we got a bang for our buck. So it was, and it was also replacing, as I mentioned before, astrocytes with good astrocytes and gray matter oligodendrocytes. Had we gone back to the beginning of the decade and said, what we're looking for, our release criteria, are motor neurons out of our cells, we would have been wrong. That would have been the wrong criteria, and we could have done it. We have a bioassay. We take the cells, we put them in the ventral horn, we get neurons. But if that were our release criteria, and we'd gone to the FDA and said, look how good we are, and this is what we're going to do every single time before we go into a patient, we would have been led down the garden path. Right now, our bioassay is making trophic factors to make motor neurons in the ventral roots, in the ventral horns, send out neurites that can be quantified. So multiple mechanisms of action are required. Now the key is, this is a regulatory nightmare. Because imagine you're going out to the FDA, and the point is, you're imputing multiple mechanisms. None of them, no one mechanism in and of itself is effective. But our standards, our regulation have to embrace that. Restoring what has already been lost. Protection and prediction hasn't really worked. You want to restore things. And in disease of aging, most of the time, these are chronic diseases. So we can try to recreate this milieu that exists in the acute environment, very metabolically active and very supportive of regeneration, I think as Steve Minger talked about in the morning. But that window is gone. That window is closed. But maybe we can recreate it here. But as I also mentioned, we need a whole new paradigm, because very, very often, it's not even just changing the milieu on a substrate that existed as it did before. The whole landscape has changed. Things have atrophied. There's been reorganization of vasculature. There's been scarring. There's been even cortical reorganization. So the approach is going to have to be multimodal. Well, that means going to the FDA for a combined product. And you heard a little bit about how this can be problematic. We used cells on a scaffold for spinal cord injury with actually fairly good response. And I won't go into that except to say that we then went to CERM to get support for this project as a disease team, meaning we want to now try to go into spinal cord injury patients. And we had the hardest time arguing with them that they kept on saying, well, how are you going to get 2 products through the FDA? It's hard enough to get 1 product, and now you're compounding your difficulty, and they wouldn't fund us. Fortunately, the scaffold now is FDA approved. The neural stem cells are FDA approved, and scaffold alone is in clinical trials. And I'm going to end with my last thing, which is replacing cells in the elderly. The caveats I think I mentioned this morning are that it's going to take very precise molding of the identity of these cells. It's no longer good enough, as I mentioned this morning, to say you're going to be a neuron. You got to say you're going to be a neuron in cortical layer 5 that's transcallosal, making contact with the thalamus and not making contact with anything else. Well, that's a lot. The derivatives are immature, which means that they may not even be primed to find the right target. And probably you're going to have to replace multiple cell types, which means multiple donor cells, perhaps in different stages of differentiation. You think that's a regulatory nightmare, trying to sell that kind of thing. It's hard, but that's probably what we need to do. It's not what Jeff Mackless and I thought in the early 90s, where you take the cell, you stick it into the damaged area, and voila, without knowing anything, the cell just becomes magically what you want it to be. These data weren't wrong. They were just naive. It was a very, very artificial situation. Now, you have to be able to say to the cell, for example, for Parkinson's disease, you got to be ventral mesencephalon, and even more, you have to be floor plate to give you exactly the A9 dopaminergic neuron. Then you can start getting some improvement in this monkey model of Parkinson's disease. I mentioned that you don't have to have a home run each time. And that actually helps you, I think, in a regulatory environment with the FDA. If you don't bite off more than you can chew, for spinal cord injury, you don't need to be running the marathon. Maybe just buying one and say, my goal is 1 or 2 segments of function, because that is good. That means that for a spinal cord injured patient, that's the difference between bladder control and not, being on a respirator or not, using your thumbs or not, being able to care for yourself instead of having your family do it or not. So that's big. And invoking some intraspinal circuitry. And then most natural diseases are not just one cell type. It's the same kind of communication. In the interest of time, I'm not going to go through the data for that, but what seems to happen is in stroke and in most diseases, the stem cell will try to reconstitute the whole area. You may want neurons. And initially, historically, this is kind of interesting. We would go into stroke, we'd put in the neural stem cells, we'd get 5% neurons, and the rest would be glial or even undifferentiated neural stem cells. And we said, oh man, we're failures. Why are we doing so badly? Until we reflected on the fact, well, that's the normal ratio in the normal developing brain. It's 5% and the most are also non-neurons. So the stem cell was doing what it knew needed to be done, and too many neurons actually probably would have been an leptogenic focus. So, and then of course, sometimes you do want glial cells, like in white matter disease, and the shiver animal is one of these that is a dysmyelinating and demyelinating disease. And then, of course, I talked to you already about ALS, where it'd be great to replace some of the motor neurons, but a bigger bang for your buck is probably replacing the chaperone cells, the astrocytes. So I'm going to end here by just ending with how I began, that even for running the regulatory gauntlet, still your most important question that you have to ask is, does the data make sense? Does it make sense based on your knowledge of the cell? And does it make biologic sense based on your knowledge of the disease? And still, the biggest obstacle, as I ended this morning's session, is we still, for most diseases, don't know what needs to be fixed. So, thank you.
Thank you, Evan. I say there are few people in the world who can speak as competently about the basic science and the regulation all at once with a fantastic clinical mindset. Evan spoke at great length about actually, understanding the biology here is a limiting factor, and we don't need to prove everything. But the reality is there are a few axioms in medicine. One is that people get ill, one is it takes money to do things, and you could argue that the third one is that if you don't take the medicine, it's probably not going to do you a lot of good. And a key thing there, obviously, is patient adherence. And yesterday we heard from Peter Nikita about risk management, and actually there are as many risks in not taking your medicine as there are in taking too much of it. So when we look at these essentially, to some extent, experimental medicines where we're learning along the way, particularly with combinational therapies, there's a huge amount of risk, there's potential benefits, and there's a lot of uncertainty. And if we're a community who are going to mitigate that uncertainty, a key part is understanding what patients are doing with their medicines. So without further ado, I think an absolute world leader in implementing patient adherence tools is Andrew Martello, a really innovative company called Spoonful of Sugar. The name is not only fantastic, but their products are as well. Andrew.
Okay, so I'm going to transport us from drug discovery, from the lab to the bedside, past the bedside. What happens when we give these medicines to patients? So I'm involved in a 15-year research program which looks at the fundamentals, why we don't take medicines, and how we can understand what we call the real determinants of medicines-related behaviour. But to set the scene, we know that non-adherence is one of the major fault lines in modern medicine. In developed countries, the WHO suggests that adherence to long-term therapies is between 30 and 50%. 20 to 30% of medication prescriptions in the US were never filled in 2013. Only 51% of Americans treated for hypertension are adherent to their long-term therapy. 25 to 50% of patients discontinue statins within 1 year of treatment initiation. And the direct cost to the US healthcare system alone, between 100 and 289 billion. So a huge, huge challenge. By 2020, the number of Americans who are going to be affected by at least one long-term condition that will require medication will grow to 157 million, twice the population of the UK. But it's not all bad news. We can improve self-management behaviours of chronic diseases. The results of these are promising, but interventions in the past have been challenging, and I'll come to that in a moment. So let's take the current zeitgeist, okay, of informed decision-making, increased personal autonomy, and control in health self-management, and this provides a solution. But does it? We're all for power to the patient, but what are the barriers, and how should we assist? Well, the first thing we need to understand is that patients aren't robots. They don't come to a medical encounter and require some sort of digital programming. They leave and they do exactly what we ask them to do. They're not a blank sheet of paper for us to write prescriptions on. They harbour sort of erroneous and mistaken beliefs about health, about illness, and about disease. And it's these that can lead to doubts and concerns that in the end will lead to non-adherence. So do you want the good news or the bad news first? Well, I'll decide. So there were a range of interventions to improve adherence over the past 15 years. A series of Cochrane systematic reviews looked at the good and the bad. So the good news, adherence can be improved. It's a changeable behaviour. However, the bad news is previous interventions we've seen predominantly across all of these studies have had disappointing effects. Short-lived, very small results. A comprehensive review by the National Institute for Health Research and NICE identified why and what we should do about it. So number one, limitations in the design of the interventions and how they were tested. So predominantly what we would do is we'd design a study and say we're going to improve adherence to this treatment. It would fail and we would never know why. We weren't looking at process variables within this environment. Secondly, and we'll come to this in a moment, is the failure to understand and address the real causes of non-adherence. I've never heard the word cool used so much at a congress. Okay, but what's cool for a scientist or what's cool for a clinician may not be perceived as cool by a patient. A lot of the content that is developed, so if you think about the patient information leaflet and those wonderful advertisements I was watching on TV today, can lead a patient to increasing doubt around their medication and not support optimal self-management and adherence. One size doesn't fit all, so we've talked an awful lot about personalised medicine today. I'm going to talk to you about personalised medicine from a behavioural, from a psychological perspective. We start to fuse those two things together much earlier in drug development so we can understand the efficacy of the medicine plus the efficacy of the behavioural support or the lens that we provide around that medicine. We're going to improve health outcomes. We're going to reduce the concerns that the regulators have and that the payers have. So I think the cartoon by Larson, Neanderthal Creativity, really describes really well the process of from lab to bedside. So exquisitely designed studies, wonderful innovations, and then we give it to the patient and it all goes wrong. So it's our thesis that the rate limiting step between innovation, medical innovation, and health gain is behaviour. The behaviour of a broad range of stakeholders. We need to create a virtual alliance of stakeholders. We need to improve and innovate our collaborations. So behaviour amongst all of these stakeholders varies, okay? And I'm going to focus on the patient later today. But if things go wrong, every single one of these stakeholders is set to lose. The clinician, if his behaviour is wrong, he's not following clinical guidelines, things aren't going to work out as well as you'd hope. From the industry perspective, they're not seeing real-world efficacy, so they'll have their trials, they'll demonstrate efficacy in their trials, and it will go wrong in a real-world setting. So we need to start to understand how to collaborate, especially within a regulatory environment, and bring these stakeholders together to develop some effective interventions because we know they're elusive. Okay, so what I want to do is focus on one human, the human right at the end of the chain. And what I want to focus on is one exemplar behaviour, whether they do or they don't take medicine. Okay, so this is what we call the perceptions and practicalities approach. It underpins the NICE guidance in the UK. It's a very simple model and explains that non-adherence can be divided into two interrelated behaviours. Okay? On your left-hand side, you've got practical barriers. This is what we call unintentional non-adherence. So the ability for the patient to be able to engage and have the capacity and the resource to engage with this treatment. But one thing we overlook an awful lot is the psychological or the perceptual side. This is what we call intentional non-adherence. And believe it or not, there are patients out there who are making conscious, if not irrational, decisions to disengage from treatment. We've developed a range of tools that help us personalise the patient experience from a psychological perspective that could be plugged in at any stage of the drug development pathway. They've currently been used widely in clinical research. At Spoonful of Sugar, we're now applying these in a digital environment, so when a patient starts treatment, we can understand their barriers and facilitators specific to their illness, representations, and their medicine that they've been prescribed. The seminal piece of work is around the Beliefs About Medicines questionnaire, developed by a chap called Professor Rob Horne, who is the Chief Scientific Officer at Spoonful of Sugar. And it's his work that has led to the development of this seminal piece of work called the Necessity Concerns Framework. Okay, so the Necessity Concerns Framework is two salient beliefs that are modifiable, that we can start to act upon within individual patient setting. So what it explains is, it's useful for explaining non-adherence, and is useful for explaining non-adherence at an individual level. So every individual that encounters a medical intervention will have their own doubts about the personal necessity of this medication relative to their concerns about taking it. Sounds awfully simple, doesn't it? But in a meta-analysis of 44 studies, nearly 12,000 patients translated into 9 countries, this model has been predictive of low adherence time and time again. I'll demonstrate that in a moment. So quite simply, patients find themselves within a necessity-concern dilemma. Okay, or we can say benefit, the necessity, versus the risk, the concern. And everybody in an encounter will start to weigh up the possibilities of both. If concerns outweigh necessity, non-adherence will occur. It will either occur because they won't start treatment, or they will discontinue or become non-adherent over time. Okay, so you're going to have to humour me for a moment now, okay? So, let's just sort of take, for example, that we all woke up this morning and we decided that we wanted to rediscover our beauty. For some of us, including myself, I'd just quite like to discover it, to be fair. I stumbled across this during my sort of research for today. And already, I'm sure some of you are making implicit benefit-risk calculations around rediscovering your beauty. So you read the first two lines, your necessity would be quite high, right? So, yeah, I want to rediscover my beauty. But then you read on, with stem cell reverse aging therapy, educated group of people, but you're going to go to the internet and just have a little dig around and look into it. All of a sudden, your concerns are starting to outweigh your necessity. Then you look, you get a free eye treatment, and it's only 169, I think. Malaysian, I don't know what the, yes. It's a Malaysian advertisement anyway. So we're all implicitly making risk-benefit calculations in terms of our encounter with medicine. So once we understand the necessity concerns profile of a patient, what we can then do is segment them based on their beliefs, okay? Their individual beliefs that are going to relate to their medicines related behavior. And I'll share a cross-sectional study that occurred in 2008. So this is the Beliefs About Medicines questionnaire in action, okay? So it was nearly 2,000 patients, 1 in 10 members of the National Association of Crohn's and Colitis. And you can see that there's a distribution of patients within those 4 segments. What this allows us to do in a real-world setting is really focus our attention on the patients that really are going to require additional support. Each individual within this segmentation strategy can receive their own personal care plan, medically, but also psychologically. And it works, okay? So if you look on the y-axis, the accepting group, much more adherent, significantly more adherent than all of the others. So the idea of any intervention that you develop for patients within an adherence support program or within a clinical trial setting when you're bringing a medicine to market is drive every patient so they sit within this accepting segment. The technology we've developed at Spoonful of Sugar now allows us to do that at an individual level. So we can find out where they are 3 months later, and continually flex the care plan for them to support optimal adherence. How do we do it? Well, interventions, as I said, are elusive, but you need to focus on 3 key aspects to optimize adherence-enhancing interventions. Failures in the past to support patients have been a one-size-fits-all approach. Now, we're hearing that the EMA are developing a pill plus as in patient information leaflet. So QR codes on boxes that take them straight to a digital source, which give them more information than that very frightening information that most of them read when they pick up a prescription. We want to take that further. We want to individualize the communication to the patient through profiling in between that process. Channels are important as well. You have to understand how to communicate with the patient, which source, and it usually needs to be a multi-channel approach. So we need to be testing these things early to reduce the risk of non-adherence. And finally, context. Where is this message coming from? Well, it's clear that the clinician needs to be involved. Every intervention that we develop is enhanced by clinician involvement. The challenges are time and training to do this. But this was a study of over 500 patients that showed that just a 12-minute telephone conversation 4 weeks after a prescription for a chronic condition reduced non-adherence in the interventional arm 9% versus 16%. And it endured, so 23% versus 34% who reported problems at 4 weeks. So what do we need to do? We need to develop an innovation plus solution. Go beyond what's in the box, okay? Because it's clear that if we carry on with our current tactic of keep doing what we're doing, we're going to fail. So we need to optimize the product. We need to understand behavior and the interface between the human and the innovation. We need to move beyond clinical variables, and it's a major mind shift, and it's going to take time to achieve. This is us, so we are a university spin-out that applies the work of the Centre for Behavioural Medicine. And if you want any more information, feel free to come and see me in the break. I'll hurry up, David, because I know you're keen to get to the panel. So thank you very much.
Thank you, Andrew, for a great presentation there. And I think that patient adherence is something we don't really hear a lot about, but really two dimensions here. One is, why don't we hear more? We're all scientists, we want to see our things being used properly. And the second component is, we all ourselves non-adhere. Today I haven't been to the gym, and as soon as this is over, I'll be going. So that's why we'll get wrapping this up quickly. But very importantly, the regulation is the narrative thread, the gatekeeper throughout all this. And whether we're looking at the basic science end and trying to get our BLA in, or IND at the other end, or whether we're looking at pharmacovigilance end at Andrew, the regulation is absolutely critical, particularly as we look at curative therapies. So here to speak about that is Bob Clay from Kynapse. Thank you, Bob.
So thank you. Won't repeat what I already said. But Evan and others have given really great insights into what goes on in a regulatory agency, what the processes are, and I hope have given you some confidence in the system that perhaps you didn't have from the popular debates. And I hope I don't erode that too much in this next part. Question, why do we regulate medicines? Well, one answer is to protect patients, and fabulous example in the previous speaker, to protect patients from adverts like the one you were just shown. Now, there's my reaction to that advert. But modern regulation is based on attempts to avoid mistakes. That's the essential foundation of the regulation of medicines, is to avoid the mistakes we've already made. These are famous errors, well known. Thalidomide is the most famous, but it repeats a pattern. And what we do is we discover ways of avoiding making that mistake again. What this does is create an environment which is described as risk-averse, or perhaps stifling innovation. And this historical perspective, for me, explains why it's difficult for the system to cope with the new avenues that you're presenting, like the ones you're presenting at this conference. The optimistic side, and one point I want to make is regulatory scientists, like the ones that Evans talked about, one of the challenges because of this history is that they weight the unknown risks, and they weight those disproportionately, I would argue, against the anticipated benefits. And one of the reasons why they weight those unknown risks highly is because they don't believe the anticipated benefits. And so you're in a conversation where you have to convince, hence mechanism of action, rational assumptions. Now, there's a positive side. FDA, other EMA, other agencies are interested in supporting innovation. Those of you who don't follow this in the detail will not necessarily have appreciated that the renewal of the FDA User Fee Act a year ago was called the Safety and Innovation Act. The European Medicines Agency has numerous activities at supporting innovation, supporting small and medium enterprises, scientific advice processes. The legislation in Japan that's been highlighted several times this week to enable stem cell therapy to be not only experimented, but also to be approved. And that's a great response. The fact that the country has a Nobel laureate in the field, perhaps explains why they noticed that they had a problem, but they've certainly addressed it. But as we've said, the regulatory system evolves in a certain way, and there are some challenges that it presents. One is I would call the problem of the average. US legislation still requires 2 adequate and well-controlled studies in order to approve a treatment. Drugs are measured. What we learn from those studies is the average effect. There have been some welcome developments like Bayesian approaches and adaptive designs, but they don't address the fundamental problem. The other area that I'd like to highlight is drugs are not safe or unsafe. They are a complex benefit of risk profiles that are more or less uncertain. And that's what the regulator's interested in. And this brings us to the point about adaptive licensing, which is an attempt to move away from this one point in time where we naively think that something is safe enough to be used to a learning environment in which we understand more deeply what's going on and how much we know at a particular time and what steps are appropriate to take. The other point that I wanted to highlight, and I heard this on the first day, and I thought it was a useful way of analyzing the question that we have, which is the regulation of these regenerative therapies, as it were. And I can't attribute this because I didn't note the speaker, but there was a model of scenarios that I think, just briefly, replacement of broken and non-functional parts was one. Intervention in pathological processes was the other. And the third, which I've reworded, but correction or mitigation of damaging environments. So if we think about regulatory pathways, which was what we were asked to think about in that context, perhaps we need to look at them for those three different areas. And also combinations of all three. The first two are pretty straightforward, I would contend, in the scope of existing frameworks for approval for drugs and devices, with the caveats that I've mentioned. It's less clear to me how the third one, fits into the regulatory pathway. I think that's much more challenging. In category one, we heard about ViaCyte, and forgive me for rushing because I do want to allow some time for discussion, but ViaCyte's an example. Replacement cells in a device, under study at the moment, makes sense. I can see how to regulate that. You can work out what it needs to do. You can do the studies in conventional trials and come to a conclusion and go to the agency and presumably get it approved. And there are many such studies, and I think the cause of optimism is the fact that there are many studies ongoing. The second category reflects a traditional, this is the pathology, traditional drug regulatory processes, identifying the pathology and intervening. Easy. Well, perhaps not so easy. But if you think about oncology treatments, that's the demonstration. Now, the challenge, however, is that the experience you've heard over the last couple of days about Alzheimer's disease really illustrates the challenge the problem has. In disease modification, notwithstanding identifying the charges, tau and others, actually demonstrating the utility of the product over an extended period of time is clearly a problem. And in fact, one example of that, I don't think even what we're doing today would have been possible had it been not for the development of adaptive trial designs that allow that to happen. And you heard an example, which is iSpy. I was involved in the pharma group that was working on adaptive trials. It took about 7 or 8 years to get to a point where there was regulatory acceptance of one necessary but not sufficient tool in the development of Alzheimer's treatments. Many other areas, and I'll leave some space. I have a couple of other examples that I'd like to mention, one of which is the novel combinations, which you've also mentioned. And here I wanted to highlight the value of another thing that's been raised, which is cooperative groups and collaborative groups that do that. And in fact, the novel combination drug guidance that was mentioned yesterday is a direct result of TB drug development. I'm part of a critical path group that we're working with FDA to develop guidelines for novel drug combinations, because if you don't do that in TB, it's going to take 25 years to develop a new regimen when we need them sooner than that. But those guidelines are applicable to everything else we're doing. So collaborative groups, cooperation is making a huge difference. And finally, just to highlight that there are many mechanisms for progressing through the regulatory procedure. But one thing that struck me based on this morning's presentation is we currently have a system that is designed to approve about 30 new products a year. It's been doing that plus or minus 10 for 40 years. The system is designed to do that. The current success rate, based on an article published by Leonard Sachs earlier this year, is 75% of things that are sent to FDA are approved. So the question I think I'd leave is, what's the transformative act that will enable us to have a system that's actually, there's nothing wrong with the system, it's just not able to cope with that future. And I think that I'll leave it there and allow some time for discussion, which is, my reckoning, 2 minutes.
Thank you, Bob.